Welcome back to the deep dive. Okay, our mission today is really to look beyond the sort of general AI buzz. We
Hmm.
want to see where it's actually becoming a vital partner in the lab, a real co-scientist.
Exactly. We're focusing on how AI is tackling the frankly brutal inefficiency in drug discovery.
Specifically, we're digging into two key challenges: finding the right biological target to begin with, and just as importantly, figuring out how to trust that the AI suggestion is actually solid.
And the snakes, well, they couldn't be higher for anyone listening who's in biotech or pharma. We know target identification is absolutely foundational. But look, up to 90% of drug candidates, they fail in clinical trials. That's a huge waste of resources and time.
90%. Wow.
Yeah, it's why companies like AstraZeneca really hammer home that right target idea.
Hmm.
If you get that wrong at the start, everything downstream is compromised.
Right. So today we're tackling this from two angles. First, we'll unpack a really fascinating recent success story. It involves AI, drug repurposing, and a new way to model liver fibrosis.
Mhm.
That shows the potential, right?
Mm.
Then we pivot to the bigger picture. How do we build, standardized tools to rigorously check all these AI models? We need confidence, not just speed.
Let's dive into that specific challenge first. Liver fibrosis. It's a major global killer. We're talking chronic injury, leading to this buildup of extracellular matrix ECM. Basically, scarring.
Scarring that destroys the liver.
Precisely. It causes about 1 million deaths globally each year. And the main culprits, these activated cells called myofibroblasts, or MyoF, they get switched on by damage signals like TGF-beta 1 and just start churning out excess collagen.
And getting drugs for this has been tough for ages. Why was that? You mentioned model systems before.
Yeah, the research was really held back by, frankly, poor models.
Oh.
Animal models often don't mimic human fibrosis accurately. What works in a mouse just fails in people. Species differences are a big hurdle.
Right.
And the older in vitro, like dish-based models, they just didn't have the right mix of human liver cells. They couldn't replicate key features, especially those thick collagen fibers you see in actual human scarred livers.
So they couldn't really model the disease properly.
Not fully, no. Which brings us to the experimental fix. These multi-lineage human hepatic organoids grown in microwells. They call them micro-HOs.
Micros. Okay, what makes them so special?
Well, they've got the essential liver cell types, hepatocytes, cholangiocytes, but the critical part. They actually reproduce the process, they form those damaging myoF cells, and they generate the thick collagen filaments when you expose them to TGF-beta.
Ah, so it's a much more human-relevant testing ground.
Exactly. It gives you a platform where you can actually see if a drug counters the human disease mechanism.
Okay, great setup. So you've got this high-fidelity model. Now, where does the AI come in, this co-scientist?
Right. So the researchers didn't just do a massive random screen. They used a specific AI system, a multi-agent one built with Gemini 2.0, designed to generate hypotheses.
Hypothesis generation, not just pattern finding.
Precisely. And they gave it a very targeted mission. Find potential anti-fibrotic drugs that work by tweaking epigenomic modifiers. Things like HDACs, DNMT1, BRD4, the machinery that controls how genes get read.
So a much more sophisticated approach than just throwing compounds at cells.
Definitely. And the results really backed up the AI's focused hypothesis. They tested 25 drugs in these micro-HOs.
Right.
5 of those showed significant anti-fibrotic effects, and they actually help promote liver regeneration. That's a key bonus.
And the AI's picks.
Here's the kicker. 2 of those 5 winners were exactly the types the AI co-scientists recommended: an HDAC inhibitor and a BRD4 inhibitor.
Wow. Okay, but wait. The source mentioned something interesting. Targets picked by humans based on, the existing literature, they didn't fare so well.
That's a crucial point, yes. Targets that looked promising based on established knowledge, specifically EZH2 and SMARCA24 inhibitors, selected by a human scientist reviewing literature, they actually failed to reduce fibrosis in this high-fidelity micro-HO platform.
So the AI, focus on epigenomics, outperformed the sort of standard scientific consensus in this specific accurate model.
It did. It suggests the older models, or maybe human bias in interpreting literature, might have pointed researchers down the wrong path previously. The AI cut through that.
And the lead candidate that emerged was?
It was the AI-recommended HDAC inhibitor, Vorinostat, also known as SAHA. And the really exciting part here is that Vorinostat is already FDA approved as an anti-cancer drug.
Ah, so repurposing potential.
Yeah.
That dramatically speeds things up, right? Less safety testing needed up front.
Exactly. It de-risks the whole process significantly.
So let's get into the mechanism. You said it's an epigenomic modifier. For someone listening, what does that actually mean it was doing inside those organoids?
Yeah, the details are pretty striking. Vorinostat essentially rewires the cell's operating system. It reduced those TGF-beta-induced changes in chromatin structure, how the DNA is packaged, by a massive 91%.
91%. So it wasn't just blocking a signal. It was fundamentally changing the cell's internal landscape.
That's the key insight. These drugs weren't just acting like a simple brake on a pathway. They were actually changing cellular differentiation. They stopped the hepatic stellate cells, the HSCs, from turning into those bad myo-F cells.
They prevented them from becoming the problem cells in the first place.
Even better, they rerouted them. The HDAC inhibitor, like Vorinostat, and another one, a GSK3 beta inhibitor, they generated what the researchers called inhibitory mesenchymal cells, or MESIN. The BRD4 inhibitor created a different type, MESWNT cells.
So it's like redirecting traffic away from a crash site.
That's a great analogy. Instead of just blocking the road, they're steering the cells onto a different, healthier path, potentially even a regenerative one. It's not just stopping disease, it's promoting a different outcome, a steering wheel, not just a brake.
That combination, the specialized AI finding the right kind of target and the high-fidelity model proving it works, that's a powerful demonstration.
It really is.
But, like you said, this success actually highlights a broader issue, doesn't it? If every team builds their own AI, their own models, how do we compare them? How do we trust them across the board? This sounds like that benchmarking gap you mentioned.
That is the critical gap, yes. You've got AI tools popping up everywhere, but no standard objective way to say, okay, this AI for predicting cancer targets is actually better than that one. Or this model works for neurological diseases, but not fibrosis.
Right. We need a common yardstick.
Exactly. So to address this, researchers developed an integrated framework. It has two parts: Target Pro, which is a specialized machine learning workflow for specific diseases, and TargetBench 1.0, which is the actual evaluation system, the benchmark.
Okay, tell me about Target Pro first. Its goal wasn't just finding any target, but predicting which ones are likely to make it into human trials, phase 1 and beyond.
Precisely. That's the key difference. It's trying to predict clinical success, not just, hitting a protein in a lab test. A much harder, much more valuable prediction.
And they trained it broadly.
Very broadly. On 38 different diseases across 5 major categories: oncology, metabolic, immune, fibrotic, and neurological diseases. That breadth is vital because, well, cancer biology is very different from Alzheimer's biology.
Makes sense. And what kind of data did Target Pro use to make these predictions? You mentioned multimodal scores.
Yeah, it's a real integration. It uses 22 different scores. It combines deep biological dataomics like gene expression, protein networks, causal links with text-based data.
Text-based, like research papers.
Exactly. Things like attention scores from scientific literature, how much grant funding a target gets, how often it's mentioned. It forces the AI to weigh the hard biology against sort of the scientific community's focus and investment.
Okay, digging into those features is interesting. The analysis using SHAP values, which show feature importance, revealed that what the AI relies on changes depending on the disease type. Why is that important for a scientist to know?
It's crucial because it proves the model isn't just using a blunt instrument. It's adapting. It shows context dependency. If the same features were important for every disease, you'd suspect the model wasn't really specialized.
So what did they find?
Well, for cancer and fibrotic diseases, the omics data, the raw biology, the genetics was most important, made up about 64–65% of the predictive power. Which makes sense, right? Cancer is driven by genomic changes.
Right. But for the others, immune, metabolic, neurological, it flipped.
Completely flipped. For those, text features became dominant. Over 50% importance for immune diseases, high 40s for metabolic and neurological. It suggests that for these complex systemic conditions, the accumulated knowledge in literature and funding trends might currently be stronger predictors of clinical progression than just the raw omics alone.
It's like using different tools for different jobs. Biology heavy for cancer, maybe more literature consensus heavy for immune disorders.
You got it. Different maps for different terrains.
Okay, so this specialized context-aware model, Target Pro, how did it stack up against the big generalist AI models we always hear about?
Right.
Like GPT-4o, Grok 3. They use TargetBench 1.0 to compare them, right?
Right.
On that clinical target retrieval metric.
Yes, and this is really the headline result. Target Pro achieved an overall clinical target retrieval rate of 71.6%.
71.6%. How does that compare?
It blows the general LLMs out of the water. It was a 2- to 3-fold improvement over the tested LLMs, which only managed retrieval rates between about 15% and 40%.
Wow, 2 to 3 times better. That's definitive.
It really is. It strongly suggests that for this kind of highly specific, high-stakes prediction in drug discovery, specialized, supervised models like Target Pro significantly outperform general-purpose AI. The generalists just aren't tuned for this task.
Okay, so specialization wins for finding targets already known to be in the clinic. But the real gold is finding new targets, right? High-quality, novel targets that won't fail later. How did Target Pro do on evaluating the quality of its new predictions?
That's where TargetBench really adds value, assessing the actionability of those novel candidates. And Target Pro excelled here too, based on factors crucial for actual drug development.
Like what? Give me an example.
Okay, take protein structure. Having the 3D crystal structure of a target protein is massively helpful for designing a drug to hit it.
Right. It's like having the blueprint for the lock you're trying to make a key for.
Exactly. Well, 95.7% of Target Pro's novel predicted targets already had an available protein crystal structure. That's huge.
Almost all of them. That saves incredible amounts of time and effort down the line.
Massive shortcut, yes. Then there's druggability. 86.5% of its novel targets were classed as druggable, with existing clinical evidence suggesting they can be targeted by drugs.
Okay, good.
And remember Vorinostat, repurposing potential. Target Pro found novel targets where 46% already had approved drugs in other diseases. That's nearly half. Compare that to 17-28% for other platforms. That's a huge de-risking factor.
So it's finding targets that are not only biologically plausible, but also structurally accessible, druggable, and potentially repurposable.
And experimentally ready, too. Target Pro's candidates had, on average, 1.4 times more available bioassays lab tests compared to other models, meaning you find a target, and chances are there are already ways to test compounds against it. They're ready for action.
So wrapping this all up, what's the main takeaway for you, the listener, from all this today?
I think the core lesson is about integration and specialization. Breakthroughs like the liver fibrosis one need that highly specialized AI, the co-scientist, focused on epigenomics. But to make AI broadly reliable and trustworthy across the industry, you need rigorous, context-specific benchmarking like TargetBench.
It's the combination that creates actionable intelligence you can actually bet R&D budgets on.
Precisely. It moves us away from just educated guesses based on past literature towards more data-driven predictions of actual clinical success.
And the ultimate vision here sounds pretty ambitious, almost like taking the human out of the loop.
Well, the ultimate goal, the provocative thought maybe, is to connect all these pieces. Imagine a fully automated, closed-loop discovery system, a self-driving lab.
Self-driving lab.
Yeah, where the specialized AI like Target Pro prioritizes targets, robotic systems automatically run the experiments, maybe using models like the micro-HOs, and the results feed directly back to retrain and improve the AI. A continuous cycle of prediction, testing, and learning.
Wow. Minimizing human bias and lag time completely.
That's the potential endgame. It's the foundation, many believe, for the next generation of drug discovery.
A truly fascinating glimpse into that future.
Yeah.
And if you listening are involved in R&D or just curious, you can actually check out TargetBench 1.0. It's public, right? You can assess your own target lists.
That's right. It's available online.
Okay, the place to go is targetbench.org. That's targetbench.or-g. Definitely worth exploring. Thank you for diving deep with us today.